Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha and in today's lecture I am going to talk to you about the tertiary structure of proteins. The tertiary structure of proteins forms the third level of protein structure. Let us see what objectives we are going to address in this lecture. First, we are going to understand the basics of the tertiary structure of protein. We will have a brief description of the structural classification, study about the factors forming and stabilizing the tertiary structure, and also know how to determine the tertiary structure of proteins. Later, we will explain in silico methodology for structure determination and the modeling of proteins. We hope you have an enjoyable time listening to this lecture. This slide gives a concept map of this module. We all know that the protein has four basic levels of structure, which is the primary structure, secondary structure, tertiary structure and quaternary structure. In this module, we are going to look at the tertiary structure of protein specifically and look at various interactions involved in tertiary folding, the process of protein folding, the techniques involved in tertiary structure prediction and in silico approach of protein modeling. Introduction. Why proteins? Proteins are functional molecules and functionality comes from its tertiary and quaternary structure. Main aim to study protein structure and function is to understand where and how protein structure and function have an impact on living endeavor. Protein structure has four levels of structural organization: primary, secondary, tertiary and quaternary. Why tertiary structure is so important? Tertiary structure of protein defines three-dimensional space orientation of an entire polypeptide. An independent folding unit of the 3D structure is called a domain. Some tertiary structures contain only one single domain known as monomeric protein, while others may contain more than one and is known as oligomeric protein. 3D structure allows us to recognize biological processes at the most ground level, like how different molecules interact in the presence of an enzyme catalysis, mode of action of drugs against diseases at the molecular level or reactions at molecular level, for example, the sickle cell behavior of red blood cells. Inside the tertiary structure, life can be defined as a synchronized interaction of proteins, and every forms of life form microorganisms to a specialized complex eukaryotic cell are based on proteins. Tertiary structure, this word defines to a geometric shape of topologically arranged various kinds of secondary structure of proteins. Various kinds of secondary structures are alpha helix, beta sheets and terms which have a capability to pack together. The figure below shows schematic representation of all levels of protein organization. Classification. The main aim of in vivo synthesis of tertiary structure in all living organisms is to promote proper functioning of a system through interaction of various macromolecules and micromolecules. On the basis of their solubility structure, tertiary structure of proteins are further classified into two large classes: fibrous proteins and globular proteins. Fibrous proteins. Fibrous proteins are insoluble and have parallel organized polypeptide chains along a single axis in the form of long fibers or large sheets. Fibrous proteins generally have a single type of motif or secondary structure. They normally associate with structural support of functions. Some useful examples of these structural elements is keratin, silk, fibroin, collagen, etc. The figure below shows silk fibroin with layers of anti-parallel beta sheets and colorized electron micrograph strands of fibroin emerging from the spinnerets of a spider. Globular proteins. Globular proteins are water-soluble proteins in nature and have a spherical or globular shape. In these proteins, non-polar and hydrophobic amino acid residues like valine, isoleucine, leucine, methionine and phenylalanine generally present in the core and initializes its folding. whereas polar residues like aspartate, glutamate, arginine, histidine, lysine are strongly concentrated on the surface and make polar proteins or water soluble proteins. Some major examples of globular proteins are myoglobin, hemoglobin, immunoglobin, enzymes, transport proteins, etc. The figure below shows a hemoglobin subunit. We hope you are finding this lecture useful. We have seen what is the tertiary structure of proteins and what is the classification of tertiary structure. The tertiary structure of protein is said to define the entire three-dimensional or 3D space orientation of an entire polypeptide. It is therefore important that this tertiary structure remains stable. Let us now dig a bit more deeper and explore what are the various forces and interactions that help in the stabilization of a tertiary structure. Interactions involved in the tertiary structure stabilization. The tertiary structure of protein is stabilized by many weak forces such as hydrogen bonds, van der Waals forces, hydrophobic interactions, ionic forces and disulfide bridges. Formation of a stable tertiary fold depends upon the interactions that differ in their relative strength in proteins. The following types of covalent and non-covalent interactions stabilize the tertiary structure of a protein: disulfide bridges, hydrogen bonds, hydrophobic interactions, van der Waals force interactions and electrostatic interactions. Disulfide bonds. Disulfide bonds are the most prominent covalent interactions in tertiary structure. Disulfide bridges form a protein fold by making a strong covalent link between cysteine through oxidation processes. A disulfide linkage cannot form between two consecutive cysteine residues, it should be separated by at least 5 other residues. Hydrophobic interactions. Non-polar substances explain to the force that leads clustering them together in order to minimize their contact with water. This self-association occurs due to hydrophobic bonds. Polar molecules in aqueous solutions interact with solvent through hydrogen bonding or ionic interactions. Non-polar molecules are incapable in hydrogen bonding or ionic interactions formations. So, when they enter the aqueous solution, water molecules attempt to build a cage-like structure around them, which reduces the overall total entropy. Hydrogen bonds. Hydrogen bonds are the major contributor in the directional stable interactions and folding of alpha helices, beta strands and tertiary proteins. Hydrogen bonds between main chain carbonyl oxygen C double bond O and amide nitrogen of NH in the hydrophobic core of the protein satisfy to hydrogen bonding potential in proteins. Hydrogen bonding between a protein and its ligands, various substrate nucleic acid proteins and inhibitors, etc. also provides specificity of interaction that is a fundamental approach of molecular recognition. Properties of hydrogen bonds. Amino acids which are participating in hydrogen bonding have the following characteristics. They may act as hydrogen bond donors like arginine and tryptophan having pi electrons of guanidine and indole group respectively. They may be both hydrogen bond donors and acceptors. Some amino acids have side chain which can donate as well as accept hydrogens like asparagines, glutamine, serine, threonine and tyrosine, but the hydrogen bonding potentiality depends on the pH of the environment. That is, tyrosine, serine and threonine are donor and acceptor in solvent system, whereas they act as donor only in the hydrophobic or non-polar region. In some amino acids, hydrogen bonding capacity is pH dependent. In this case, same amino acid may act as a donor and acceptor in certain pH range, but turn up different in another pH range. For example, aspartic acid, glutamic acid and histidines are pH dependent. Hydrogen donor and acceptor in different amino acids. In all cases, hydrogen bond involves an electropositive atom, donor, and an electronegative atom acceptor with a range length from 0.26 to 0.34 nanometers. Instead of this, various another types of hydrogen bonds are involved in protein folding, sharing donor and acceptor atoms within the side chains. Van der Waals force of interaction. Van der Waals forces are weak attractive forces between atoms or groups towards another within the same molecular entities. They take place between induced or permanent dipoles. There are three types of terms includes for VDW or Van der Waals force interaction: dipole-dipole interaction, dipole-induced dipole interaction, and induced dipole-induced dipole interaction. Dipole-dipole interaction. This kind of force generates between molecules containing electronegative atom, which forces molecules to orient them so that the positively charged end of one molecule is oriented towards negatively charged end of another. Dipole-induced dipole interaction is a permanent dipole which has a tendency to induce transient kind of dipole in a nearby molecule through distorting its electronic distribution. In induced dipole interaction, motion of electrons in one atom affects to another atom electron's motion. At any point when more electron may be present on one side of the nucleus than another. So, atom becomes small, and at that time one side shows a partial negative charge and another side shows a partial positive charge. Therefore, a transient type of dipole in one molecule polarizes to neighboring molecule's electron. This slide gives a pictorial understanding of the various type of Van der Waals interactions we just studied. A shows dipole-dipole interaction, B shows dipole-induced dipole interaction, and C shows induced dipole-induced dipole interaction. Electrostatic interactions. In proteins, electrostatic interactions occur between positively charged amino acid residues of side chain as well as in between carboxyl and amino groups of polypeptide ends. Amino acid residues like lysine, arginine, histidine with positive charge show interaction with negatively charged amino acid residues like aspartate and glutamate. In electrostatic interaction, Coulomb's law explains the potential energy involved between the two opposite charges. It is given as V is equals to Q1 Q2 divided by epsilon 4 pi r square, where Q1 and Q2 are the magnitude of opposite charges, r represents distance between these two opposite charges, and epsilon is the permittivity of the medium. So, students, we saw that the disulfide bridges, hydrophobic interactions, hydrogen bonds, Van der Waals force interactions and electrostatic interactions are some of the key interactive forces that allow the stabilization and folding of the tertiary structure. We looked at these forces in detail in the previous section. In the next section, we are going to see how a protein with a primary and secondary structure folds to form a tertiary structure and what are the underlying mechanisms behind it. We will also look at some important techniques and approaches to decipher and interpret the tertiary structure in proteins, for example, X-ray crystallography, NMR spectroscopy, 2D NMR and some bioinformatic tools. Let us take a look at these. Process of tertiary structure formation. Protein folding is a physical process by which a polypeptide folds into a functional conformation. In general, protein folding starts from the local segments like alpha helix and beta sheet interactions. The process is extremely rapid in its initial steps. Secondary structure in native protein forms within 5 minutes which initiated protein in folding. Initiation of protein folding takes place from compact hydrophobic cores, consists of nonpolar residues. Those next to lead a rapid spontaneous collapse. Folding mediated by molecular chaperones. All types of proteins cannot fold spontaneously after synthesis. Hence, in vivo folding of many proteins needs specific kinds of assistance of molecular chaperones. During the synthesis of nascent polypeptide chain, folding is initialized by molecular chaperones to prevent formation of incorrect structures. This phenomenon was first observed in E. coli, and their concentration increases at high temperature or at heat shock, therefore also known as heat shock proteins or HSP. Both prokaryotes and eukaryotes contain the HSP70 family, that is the 70 kilodalton proteins. Members of HSP70 bind to hydrophobic amino acid region on string of newly synthesized polypeptide and guides its proper folding. In contrast, another heat shock protein family HSP60 bind to partially folded or incorrectly folded protein to create a favorable environment for refolding of the protein. This slide shows a graphical presentation of the process of protein folding through chaperones. Proteins to domain organization in tertiary structures. The tertiary structure of protein contains more than 150 residues, which may have more than one structural unit known as domains. In simple way, domain may be described as a region of a protein that shows structural homology with other proteins. Stability of domain structure is also governed by same kind of interaction which have been described in earlier sections. All domain structure of protein may be further organized into three major classes: domains with major content alpha helices, domains with major content of beta strands, and domains with a mixture of alpha helices and beta strands as a major content. Gene duplication and fusion is one of the major factors for domain formation. So, as a result, if a domain is added into another protein, it may lead to some new and additional properties. Different types of domains in proteins. There are different types of domains in proteins. Bromodomain from Saccharomyces cerevisiae PDB ID 1B91 contains the helix-turn helix domain. SH3 domain, alpha spectrin from chicken PDB ID 1 SHG, a beta domain, and acyl phosphatase PDB ID 2 ACY mixed alpha beta protein containing 5-stranded twisted beta sheet. Interpretation of tertiary structure. There are various biophysical and computational methods available to deduce tertiary structure of proteins like X-ray crystallography and nuclear magnetic resonance. Most of the known structure of protein are deposited into the protein data bank, which can be easily accessed via the internet. Structure determination through X-ray crystallography. X-ray crystallography is one of the most effective approach for macromolecular structure analysis. X-rays are electromagnetic radiation with wavelength range of about 0.02 angstrom and 100 angstrom. When beam of X-rays is thrown over a crystal of the protein, part of the beam passes straight through the crystal and rest of it is scattered in the different direction. These scattered of diffracted beam detected by X-ray films. The intensity of the scattered beam is proportional to the blackening of the film and measured by solid state electronic detector. The amplitude of the wave scattered is proportional to the number of electrons of an atom. The scattered rays has a tendency to cancel or reinforce each other if they are in out of phase or in phase. The resulting diffraction pattern is dependent on atom arrangement. X-ray crystallography. This slide shows different steps in the determination of the structure of protein by X-ray crystallography. A. Crystal of protein. B. X-ray diffraction patterns generated from the crystal. C. Electron density map of the protein. D. Regions of highest electron density reveal the location of the atomic nuclei, and on the basis of these nuclei, final tertiary structure is deduced through Fourier transform. E. The completed structure of the protein. Structure determination through nuclear magnetic resonance. Another method for prediction of protein structure at the molecular level is NMR. In NMR technique, radio waves are applied on protein solution under high magnetic field, then frequency of the relaxation of proton is measured after their excitement by the radio frequencies. But very few isotopes show this kind of intrinsic magnetic property. In these atoms, protons under high magnetic field resonate in between two spin states called the alpha and the beta and provide the frequency corresponding to the energy difference between the alpha and the beta state. Therefore, by varying the magnetic field, a resonance spectrum of the molecule is obtained. Through this spectrum, chemical environment around the nucleus can be deduced through a chemical shift. The movement of electron around the nucleus generates a local small magnetic field which opposes the external applied field. This kind of shielding depends on the electron density of surrounding. Therefore, nuclei will feel different fields and resonate at different degrees. This phenomenon is known as chemical shift relative to a standard compound, that is tetramethylsilane. Two-dimensional NMR. In-depth information of atoms can be achieved by examining how the spins of different protons affect their neighbors. Effect of altering the spin of one nucleus on the spin of the neighboring nucleus by inducing a transient magnetization can be used for detecting the location of atoms relative to one another in the three-dimensional structure of the protein. Within this method, a two-dimensional spectrum is obtained by a nuclear Overhauser enhancement spectroscopy, also abbreviated as NOESY or NOESY. Tertiary structure assessment with bioinformatic tools. Till 2014, about 22,000 sets of atomic coordinates were deposited in protein databases (PDB), and this value deposition of new structure is increasing every day. Currently, PDB has a total of 35,350 sets of atomic coordinates. PDB database can be easily accessed through the website provided. Through computational method, comparative or homology modeling of the structure of unknown protein can be made through known or homolog template. Students, we have now come to the end of this lecture. Thank you for being there with us. Let us quickly run through what we have learnt in this lecture today. Proteins are the functional molecules, and this functionality comes from its tertiary and quaternary structure. Tertiary structure of protein defines the three-dimensional space orientation of an entire polypeptide. An independent folding unit of the 3D structure is called a domain. On the basis of their solubility structure, the tertiary structure of proteins are further classified into two large classes, the fibrous proteins and the globular proteins. The tertiary structure of protein is stabilized by many weak forces such as the hydrogen bonds, van der Waals forces, hydrophobic interactions, ionic forces and disulfide bridges. Protein folding is a physical process by which a polypeptide folds into its functional conformation. All types of proteins cannot fold spontaneously after synthesis. Hence, in vivo folding of many proteins needs specific kind of assistance like those of molecular chaperones. The various biophysical and computational methods which have been available to deduce tertiary structure of proteins include X-ray crystallography and nuclear magnetic resonance. Thank you for watching this lecture on tertiary structure of proteins.